Yes,
and we explained about the different bonds inside the chain, yeah? Am I right? Now, I want to tell you about arranging different types of amino acids in peptide chain. At first, guys, I told you that we have some polar amino acids and some non-polar amino acids. Am I right? Here, the greens are non-polar amino acids with non-polar side chains. And the blue ones are amino acids with polar chain. Imagine you are the signature of Mason and Ahmed. Okay. Excuse me for erasing that. Okay, guys, imagine you have a peptide chain and you wanna put it in a hydrophobic area, hydrophobic environment or moiety. Like, for example, some place with different lipids. What is happening for this peptide chain? How? Which amino acids come to the surface and which of them are in the pole?
Polar outside?
In hydrophobic area.
Outside and hydrophobic parts inside.
Hydrophobic outside.
No, hydrophilic inside.
Hydrophilic
inside.
You forget about this slide, okay? I want to tell you, we are put our peptide chain in the hydrophobic area, environment or moiety. The
outside.
polar or non-polar?
Non-polar.
Non-polar is outside and polar is inside. But here, this is another situation. It is in a polar area, like water moiety, like our blood circulation. What is happening? Base of our blood circulation is water. Am I right? In those areas, the hydrophilic or polar amino acids come to surface and the core part of amino acid will be non-polar amino acids. Okay? Did you get the point? Another example that I want to tell you is about the cell membrane. Different membrane, cell membrane and organelle membrane. You can see that hydrophobic amino acids arrange in a way that be in touch with hydrophobic parts of cell membrane and lipids. These are, do you know what are these? Phospholipids. These are polar part, polar head, and here is non-polar tail. Am I right? The amino acid arrange in the way that polar parts be in touch with polar area, excuse me, and non-polar amino acid be in touch with non-polar area and objects. That was related to the arranging of amino acid in tertiary structure. And lots of our protein in our body, like albumin, like fibrinogen. Salam alaikum. You were killing him in the laboratory.
Yes.
Yeah. Why you were killing him? Do you know why? He told him that Yeah. You are not scientists that you want to kill your friend without hurt yourself. Why don't you mix them with each other? Okay, just kidding.
It's the point.
Guys, it was about tertiary structure. For example, the thing that I told you, this structure is correct about alveoli, about globuli, about spherical proteins that we have. Okay? It was about the arrangement of different amino acids in different areas. Another hierarchical structure of proteins is quaternary structure that we have in situation that we have more than 2 monomers, at least 2 monomers. You need 2 subunits to say that I have quaternary structure. Other than these are not quaternary structure. This is hemoglobin that have alpha globin chain, beta globin chain, and heme. In the center of heme we have iron. Exactly. There are guys, except the interactions that occur in each monomer, different interactions between other subunits we have too. Okay? We have different interactions here. Guys, let me explain about physiochemical properties of amino acid. One important thing that today you do that experiment was about denaturation and renaturation. What is denaturation and renaturation?
Breaking the bonds between protein.
Which one?
The bonds are
The hydrogen. No, the bonds that hold the protein together.
Yes.
Like the weak hydrophobic interaction, the ionic bonds, the
Disulfide
bonds. When they break, the active site gets a conformational change and the substrate cannot bind to it, hence it is called as denatured.
All of you were correct. Even the friend that say we are breaking the peptide bond. It depends, guys. Guys, if the denaturation is reversible, we just, as our friends explained, we have actually breaking down the interactions inside the molecule, not about peptide bonds. What if our denaturation B is reversible? The peptide bond is broken down. The thing that today we add acid, some powerful acid, and we denature our proteins in an irreversible way. And if the thing that can make protein denatured or temperature acid-base, today we work with acid. During the denaturation, at first, we lose the secondary and tertiary structure. As our friend mentioned, we have loss of function. For example, imagine you have an enzyme that do For this protein. Just guys, do you know about protein classification? You know, I'm sure that you know.
One single complex one. There is no single one complex.
Hmm?
There is no structure or function.
Shape, structure, function.
Yes, we know it's like about the structural proteins and the globular proteins.
Yeah. Yeah, some of them are globular, and some of them are
Fibrous.
fibrous, globular, and some of them are enzyme. Can be enzyme. Some of them, guys, if I would tell you that some of proteins are simple, some of them are complex, what do I mean? Simple protein and complex protein, or heteroprotein.
Having the same one
Amino
as
acids.
simple.
Hmm?
Having the same one as the supplements.
Somehow,
Is that simple?
yeah, but how? For example, imagine I'm telling you that this is simple protein and this is complex protein. And another name, homo protein and hetero protein. What did I mean? I mean that here just we have protein subunits, yes. But here, except protein, we have other non-protein parts like lipid, like carbohydrate, like heme, like nucleotide. As you can see here, simple or homo protein, we have just amino acids, nothing wrong. Like globular, alveoli, and globulin, and fibrillar protein that can be soluble and insoluble. Soluble as fibrinogen, myosin, and insoluble as collagen and keratin. About complex and heteroproteins, we can have hemoproteins or chromoproteins. Like, no, hemoprotein, hemoglobin, myoglobin, okay, that they have heme. Lipoproteins like, do you know about them? Somehow, but we have some typiques lipoprotein. Have you ever heard about good lipids and bad lipids? LDL and HDL?
Yes.
Yeah? They are lipoprotein. Yeah, exactly. Low density lipoprotein, high density lipoprotein, HDL, LDL. I think Dr. Khalidi will say you. Okay? We'll present these subject to. And you've studied about glycoproteins, am I right? The proteins that have carbohydrates inside that, or maybe beside that. And nucleoproteins is like the histone structure that have protein part that DNA is ground it and somehow coil it. Okay? These are nucleoproteins. And we have phosphoproteins that, do you remember which amino acid was important for phosphoproteins? Becomes which amino acid starts to be phosphorylated? Exactly. Serine, exactly. Good job. This was about the classification of proteins. Guys, about simple or homo protein. Guys, do you know?
We should memorize this.
No. Just the thing that I want to tell you. Guys, I want to ask you one question. Just you are asking about exam questions, final questions, yeah? All of the time?
Because I'm so nervous about passing.
Is you are worried about, no, you will have good grade, good marks, not just passing. All of you are smart students and you can do that. Guys, you can see see here. Hemoprotein. Homoprotein. What are these? Okay. And what about homo? Yeah. Homo. Another thing, have you ever heard about homostasis or hemostasis?
The maintenance of constant internal environment despite fluctuations in the external.
Okay, balance in our body. What about hemostasis?
level of iron.
No.
Is that all blood?
Which part of blood? Yeah. Right. Hemostasis is related to blood coagulation and blood and keratolysis. For example, you have your finger injured. Okay? At first, the clot should be activated and inhibit bleeding. Okay? After a while that here is healed, the clot should be dissolved. It couldn't be here anymore. And this process is hemostasis. Okay? This was another extra point.
Extra should not be in the exam.
I will keep all of you all the time. I'm worried about the exam.
It's also related to protein.
Do you want just give you the questions and come? Yeah.
Yeah, of course.
I will just tell. I told to my friends tomorrow my flight is sooner than normal. It was for 2, and now they told me I should leave here before 11. And I'm not worried about that. Just I will give you 6 questions, and I will go in the morning. And even we don't have any class. Okay?
Are those exam questions? Exam questions or just
It was serious.
No, they are the exact questions, not examples. Do you want any more? Do you need any more? I'm here.
The solution.
I will give you tomorrow. I will no other important point to you. But just I will tell you all of this part are important, and you should study all of them one by one.
No, seriously, we want us as example, home example to be
I'm saying that.
A mock, like a mock test.
No.
You can see my paper exams.
I'm telling you, for example, if I ask you which of amino acids have essential or in disulfide bond, what is that system? I'm telling you these some questions. Just need to say all of 4 options. I'm disappointed. I will kill myself. Not with you.
Yeah, but it's excuse us. This is the very first term for us. That's why we are a bit nervous.
No,
It's okay.
there is no worry. We will pass all of them. Guys, at first, I want to tell you about alveolar proteins. There are different classes of alveolins. Alpha, beta 1, beta 2, gamma. It is not important to know, but about alveolin. Alveolin is so important, and now I want to tell you about, guys, do you know that the albumin, where is synthesized? In which organ in our body? Albumin is synthesized. Liver. Liver is the place that albumin starts synthesis. And in liver abnormalities and liver diseases, the synthesis of that decreases. Okay? And another thing, guys, 20%, 25% of total proteins contain of albumin, and in liver diseases, albumin decrease, molecules, like hormones, like fatty acids, like bilirubin, like thyroid hormones, and not something that can carry by albumin. Guys, we have a term that everything that is not soluble in our blood should be carried by someone, by something. And the general transporter in our body is albumin. Everything, for example, thyroid hormone, steroid hormone, bilirubin, all of them have their special transporter. But albumin works as a general transporter in our body. Okay? But another important thing that about albumin, it is good to know and it is necessary to know, is about negative charge of albumin because of acidic amino acids. Another thing, guys, albumin have role in regulation of oncotic pressure. Do you know about oncotic pressure?
Yes.
About cancer? No, not about oncoprotein or oncogene. Oncotic pressure, it is a physiologic situation. We have in cancer, we have oncogenes, oncoproteins. Am I right?
Yes.
Have you ever heard about them? These are the genes that cause cancer if the mutation occurs. For example, something like P53 and some other genes. But no, it is about oncotic pressure. Have you ever heard about osmotic pressure?
Yes.
What is it related to?
Salt concentration. Salt and water.
Salt and water. Salt and water, especially water. It depends. For example, here you have, for example, here, salt, for example, and water. Am I right? Here, the place that have higher concentration of salts absorb more water from the less concentrated area. Am I right? This is osmotic pressure. But guys, about oncotic pressures are related to protein. For example, albumin in your blood circulation provide an oncotic pressure. Okay? What did I mean? Imagine these are albumins. Okay? The yellow circles are sodium. I'm not speaking about them. And these are H2O or water. Guys, imagine these albumin cause absorbing some water to the blood circulation. Okay? If the albumin amount decrease, what is happening?
More
More H2O,
More
gets
H2O.
absorbed and more energy.
Inside the arteries?
More albumin comes to the blood.
No, the albumin concentration in the blood decrease. For example, here, instead of 4 albumin, you have 2. Lose 2 of them. Just have these 2. What is happening for water?
Increase. Water outside the increase.
Yeah. Yeah. Exiting of water molecules increase. And here's the place that we tell that oncotic pressure decrease. And we have a phenomenon on the name edema.
Accumulation of water in the extracellular space.
Did you get the point? It was related to oncotic pressure. When we have hypoproteinemia in different situations, we will face with edema. excretion from our kidneys. Am I right? In nephrotic syndrome, or maybe in extreme exercise, or in malnutrition and burning, in this situation, we will face, because of albumin, decreasing albumin, we have edema. If you have pressure on the different parts, that you can see edema, your fingerprint or maybe your finger space will remain in that area. Did you get the point?
Yes.
About fibrillary protein, guys, before I explain about all of them, but I think it is a little difficult, and I omit these parts, and just I will explain you about collagen and keratin, that they are insoluble. Okay? These proteins, this was related to coagulation system. At first it is soluble, and after that it breaking down, convert to fibrin is insoluble and can produce clot. Okay? The other one are proteins that are involved in movement, but these are a structural protein. Okay? In the pores, for example, in the skin, in the nail, in different parts of our body. Let's go to the hair salon at first. Okay? I want to tell you about the chemistry of curly hair and straight hair. How? Guys, our hair structure is based on disulfide bond. You can see here
Yes.
in keratin, that is full of cysteine. In the hair salon, for example, a lady come here and say that I want to curly hair. What is happening? For 6 months. Okay, what is happening? At first, they use reducing agents. What reducing agent do?
They remove hydrogen.
They remove the electrons, basically.
Separate the hydrogen from sulfur.
And hydrogen. And these disulfide bonds convert to thiol groups. And after that, they curl the hair with different instruments. Okay, they curl the hair, for example, around this structure. We told it in Persian bigudi. I don't know what are you saying in these structures that they hair around that. Okay, and after that, the hair become curl and they oxidize again. Some disulfide bonds become oxidized and they arrange in an irregular form. For example, with this disulfide bond, instead of attaching here, attached here. Because of the curling that you did. Okay, and in this area we have curly hair because of disulfide bond. Am I right? And for straight hair that we have something like keratinization.
Yes.
Do you have in your country?
Yes.
Or just in Iran?
No.
They have. They just reduce. And have you ever heard that you shouldn't use shampoos with sulfate?
Yes.
Yeah? This is because of that. Because the sulfate maybe again oxidize these structures and your hair structure become like before. Okay? That's what about curly hair and color.
After adding reducing agents, the disulfide bonds convert to what?
Here, thiol. Thiol.
Okay.
SH
Yes.
of cysteine is thiol. Okay? Another thing, guys, I want to tell you about collagen. Collagen structure and production is so complicated. Okay? It's so complicated and I don't want to concentrate on the production of that. I just want to know you some important points, including about the different collagens. It's not important about hydroxyl group are so important. And I want to tell you, guys, we have translation, and we have a protein, for example, with this structure. Okay? After the translation, these proteins have no function. Some post-translational modification should occur. Do you know what is it?
Yes, the exons and introns, the thing.
From translation to translation is about exon and intron.
Oh.
Here, for example, these elementary part, these primary part of protein maybe break down, and these protein become mature. Okay, this is post-translational modification, or maybe some, for example, hydroxyl group will add to some amino acids here. These are post-translational modification. There is no hydroxylizing in your translation. You have no code for hydroxylizing. Do you have? After translation, these modifications occur. Did you get the point? No? Again? Again? Guys, for example, you had some codon that they translate to arginine, lysine, tryptophan, proline, am I right? These are in your protein amino acid chain, or maybe they fall. But you don't have codon for hydroxyproline. Do you have? Or hydroxylizine. Okay, these changes occur after translation.
Oh, okay, so that's why it's called post-translation.
Post-translational modification that occurs most of them in Golgi apparatus. Okay? After ribosome and graph in the plasmic reticulum, lots of them occur in Golgi apparatus. Did you get the point? This was about post-translational modification, and this is the reason that collagen have a compact structure and have different H bonds between OH groups. Okay? This was about collagen. And here are some, guys, do you know? I'm worried about become confused. Collagen, co-collagen. Guys, let me tell you about other post-translational modification. At first, we have something under the name pre-porocollagen. Okay? After that, this pre eliminates. For example, here this is polypeptide that we call it pre-procollagen. At first, this part is breakdown. Here is procollagen. It doesn't have function. Again, in another process that we call the post-translational modification, this procollagen is eliminated again. And here we have a mature collagen with function. It is another post-translational modification that different parts of immature protein should break down to convert a mature protein. Mature, yeah, can have function. Okay?
Yes.
Did you get the point? I hope. Guys, I want to see you about another abnormalities under the name scurvy. Have you ever heard about where? Where? What? How?
Deficiency
Vitamin C.
deficiency of
Vitamin C.
vitamin C
And when it was described?
When, like in olden times when the sailors used to go, right, they wouldn't take fish. They would only eat meat, which would, I mean, that's what we've been taught.
Conservative meat.
Yeah.
Some conservatives.
Yeah, so they could not get vitamin C and all the vitamins in fruits. So their teeth
Fresh fruits
Yeah.
and fresh vegetables, okay?
So their teeth used to get very
Yeah.
weak and bleeding gums and everything.
Exactly. Guys, and have you ever heard about clinical trials?
Yes, clinical trials.
Clinical trials in medical, for example, you are our future medical doctor, inshallah. For example, you want to prove that this drug and this method is effective in these patient groups. At first, you should work on the cell lines, after that on animals, after that on human. In human, we call it clinical trials. For example, you give to some group of patients special drugs, for example, vitamin C, and give to another group nothing. This is clinical trial and evaluate the disease stage in different groups. The first clinical trial was for discovering the vitamin C deficiency in scurvy. Okay, what is scurvy? Guys, I told you that in collagen structure we have proline, that it should convert to hydroxyproline with proline hydroxylase. Okay, guys, the cofactor of proline hydroxylase is ascorbate or vitamin C. Guys, there is no difference if ascorbate is deficient or proline hydroxylase is deficient. This enzyme won't work, and the proline hydroxylation decrease. What is happening? Guys, in this situation, when this situation occurred, the collagen structure wasn't normal. Okay? What is happening? We will have some ecchymosis on the limbs. For example, in the hands, in the legs, there are some ptosis and ecchymosis because of fragility of capillaries. Capillary wall is full of collagen. I told you it is a structural protein. Okay? Because of deficiency in collagen, the capillary starts to breaking. They are fragile. And after that, this blood will penetrate under the skin. Okay? And we have ecchymosis. Gum disease, loosening of teeth, and poor wound healing. You can see here some ecchymosis. Do you know? It can occur after collagen deficiency. Another situation, guys, here you can see again ecchymosis in the leg. This is in the leg area. You can see these areas that because of capillary fragility. The blood penetrates under the skin. Okay? And here we have gum disease and losing teeth because of collagenopathy. Another inherited collagenopathy that I want to tell you is about osteogenesis imperfecta. It is a sonography picture of an infant in the mother's uterus. You can see here that the osteogenesis was imperfect, was incomplete. In lots of situations, it is lethal situation. The baby won't be alive anymore and will die. Here you can see Ehlers-Danlos syndrome, Ehlers-Danlos syndrome, that the skin can stretch and can have flexibility, not about our skins. Guys, what do you think? These abnormalities is just related to the bones or are just related to the skin? in lots of our parts of our body have collagen. Joints, eyes, teeth, gum, skin,
Nail.
hair, everything will be abnormal. They don't have normal life. And some of them are little and some of them have lots of complications. Guys, another categorization is based on complex protein or hemoproteins. Do you want to have a rest? Stand up, have a cup of coffee or a cup of water, a glass of water, and come back.
Just 10 minutes.
10
No,
minutes.
no, 3 o'clock.
10 minutes, right?
Okay.
No, it's not. About hemoglobin saturation, it is a little difficult and I need to explain you more. Again, this. Some complex proteins. At first, I want to tell you about hemoglobin and myoglobin that we call them hemoprotein or metalloprotein because of iron, yeah, metal. They are oxygen transporter, maybe in tissues as myoglobin or in blood circulation as hemoglobin. Myoglobin is an intracellular transporter in muscle, and hemoglobin is an extracellular transporter of oxygen in blood circulation. This was about chromoprotein. Here you can see myoglobin that they are monomer. We can see them in heart and skeletal muscles. They act as oxygen reservoir and oxygen carrier, and they increase the oxygen transportation in muscle cells. And the structure that I show you, that the non-polar amino acids was in the core and the polar amino acids was in the surface, it was correct about albumin and also about myoglobin. And it is the thing that I was telling you. Guys, this is a myoglobin structure. If I want to say you by detail, each of these parts have different roles. And the heme interaction with globin chain is so complicated. I omit those parts and just concentrate on important issues. Here you can see heme. Guys, we have a protein part and another non-protein part. I will tell you in enzyme, we call the other non-protein part that attached tightly to protein part, we call it a prosthetic group. Have you ever heard about that? In which subject? Biology. Prosthetic group. That here, heme is a prosthetic group that made from iron and tetrapyrrole ring. You will study next semester, inshallah, about the tetrapyrrole structure. Guys, protein part of hemoglobin have different globin chain. It can be alpha, beta, gamma, zeta, epsilon, and delta. And the prosthetic group is Fe, 2 plus, and tetrapyrrole. Could be Fe3 plus in hemoglobin? It's not correct, and it's in some disease condition that we call it methemoglobin, but about normal hemoglobin just we have Fe2 plus. Guys, here you can see tetrapyrrole ring of heme, and here is iron, ferrous iron, and we call it porphyrin. That attach here, guys, by histidine. It's not important to memorize. Just know these are 4 subunits of hemoglobin. Guys, in hemoglobin have a tetramer structure, 4 chains. Alpha, beta, 1 dimer, and alpha beta 2 dimer. Guys, what does dimer mean? Two monomers. They are dimer. Abdullah, Safar, Maria, and all of you are present in. Okay, no, just I put empty. It was about hemoglobin that we have 2 alpha beta subunits, and about where are they in RBCs or red blood cells, and they can transport oxygen from lung to the tissue, and in the tissue transfer carbon dioxide to the lung. I will explain you completely. Guys, normal concentration of hemoglobin in blood is 12 to 14 grams per deciliter. If our hemoglobin level be less than this, we have which disease? Anemia. Okay? Anemia. This level is so important. Guys, have you ever seen or have you ever heard about different hemoglobin types? Yeah? Could you tell me about hemoglobin A is the normal hemoglobin in adults? And hemoglobin A2 is another hemoglobin in adults that the amount of that is not high, 2 to 3% of all our hemoglobin. Guys, in one situation it increased. Do you know about that situation?
When we go from low height to higher?
Maybe, but no, about pathological situation I'm telling you.
Cosmic.
In one situation, one pathological situation, we have increasing of hemoglobin A2 because the synthesis of hemoglobin A decreased. Do you know what is that condition?
It's about feedback.
No. Have you ever heard about thalassemia?
Yes.
Yes. In thalassemia, I will explain you. The synthesis of one of these chains decreased. And for compensation, the synthesis of these increase. And the increasing of that is an indicator of thalassemia. Okay? I will tell you. Guys, we have hemoglobin F. Do you know when we have this hemoglobin?
Yes.
Sorry?
When we are in the uterus, we are fetal stage.
Yeah, in fetus. When we have hemoglobin F, before our birth, our prominent hemoglobin is hemoglobin F. After that, they convert to hemoglobin F. There are different classes of hemoglobin during infancy, from the first week of our life till when we have birth. And guys, have you ever heard about hemoglobin A1C?
Yeah, HbA1c, yes.
When?
Diabetes.
Exactly, guys. Do you longer time. And what is happening? This hemoglobin binds to sugars. In normal situation, the hemoglobin that's attached to sugar is normal 4 to 6%. But in diabetic situation, when uncontrolled diabetic situation, we have some diabetic patients that their hemoglobin A1C is less than 6. It indicates that that person was controlled diabetes. He or his or she, her or his diabetes was controlled. But in uncontrolled situation, the blood glucose increase and it can't decrease till 140 milligram per deciliter. That's why the hemoglobin plays with more sugar. And the amount of that increase in blood circulation. So, hemoglobin A1C is important for monitoring diabetic patients. Okay? This is for monitoring diabetic patients. For example, you have diabetes and eat everything. Colas, rice, every carbohydrate product, and just before your test night, for example, before referring to the laboratory, you start to have suitable diet. And your fasting blood sugar become near normal. But this hemoglobin A1C will indicate to your doctor that you weren't in a good diet, in a good situation. And for example, the doctor said, no, maybe you have good diet, but your body can't work effectively. And your doctor understands that should decrease or increase your drug dose. Change your tablet, change your drug. For example, start administrating insulin. Okay, it was for diabetic patients monitoring. Okay? Guys, another thing that I want to tell you is about hemoglobin structure. Here we have alpha beta 1 and alpha beta 2. They are attached to each other with strong interactions. These alpha beta and these alpha beta, they are strongly gathered together. But about these two dimers, there are some weak interactions. What is happening? The thing that happened here, when oxygen comes to the molecule, these weak interactions start to breaking down. We say that intense or tight structure, we have the oxyhemoglobin. What do I mean? Imagine, this is T in English. Am I right? Am I in the shape of T? I don't want accept no oxygen. Tense form is deoxyhemoglobin. I don't accept hemoglobin because my hand is closed. But in relaxed form, my hand is open and I can accept hemoglobin. And I'm in relaxed form. Be careful. If I ask you that T form of hemoglobin, be aware that it is deoxyhemoglobin. And R form is oxyhemoglobin. Okay? In oxyhemoglobin, the weak bonds start to breaking down and the conformational changes occur and the place for oxygen start being open and oxygen comes to the structure. Did you get the point?
Yes.
I want to tell you guys, have you ever seen this pulse oximeter?
Yes.
In COVID, yeah, we have exactly something like this, exactly something like this. Guys, it is based on light absorbance, differences in light absorbance between oxyhemoglobin and deoxyhemoglobin. The light absorbance of these hemoglobin are based on these lights that they actually start enter to this area, they can evaluate the oxygen saturation. Guys, I want to tell you, at first, when one oxygen, we have 4 oxygens here. Am I right? Yeah?
Yeah.
When the first oxygen one comes here, it is difficult.
Yes.
It is-
has
Yeah, yeah. At first, it has low affinity. After coming the first oxygen, some conformational changes occur. Allosteric effect, yeah. And the other parts attached to oxygen easier than the first. We call that the cooperative effect. Cooperative effect. This is the effect of protein with more than one subunit. Okay?
Yes.
Some enzymes or some molecules that have more than one subunit, they have cooperative properties. One structure, one subunit helps the others to work more efficiently. Okay? That was about pulse oximeter. That now I want to tell you about, guys, this is, we call the oxygen dissociation curve. What does it mean? Here you can see the saturation of hemoglobin by oxygen. Okay? And here we have partial pressure of oxygen. You can see that myoglobin have a hyperbolic curve. What did I mean? I mean that myoglobin attached to oxygen easier than hemoglobin and release that harder than hemoglobin. On the other hand, hemoglobin at first attached to oxygen harder than myoglobin, and after that suddenly you can see high affinity because of cooperative effect. Guys, here we have a term under the name P50. This is the pressure of oxygen that the hemoglobin is saturated by 50% of its capacity. For example, the hemoglobin is 2 parts of that is saturated with oxygen. The 2 parts of that is empty. This pressure for myoglobin is near 1 millimeter Hg. You can see 50% of myoglobin is saturated, and for hemoglobin it is near to 26 millimeter Hg. I will explain you more. These are some explanation for you. Myoglobin has higher affinity to oxygen, and guys, this higher affinity, do you know what? I told you, cooperate, okay. Guys, in the lungs, we have high pressure of oxygen, am I right? In this place, hemoglobin can be saturated with oxygen, am I right? And on the tissues, we have high metabolism rate.
So it needs oxygen.
What do we have?
It has less oxygen, so it needs oxygen from the hemoglobin.
Yeah, we have decreasing oxygen and we have increasing what? Carbon dioxide. And on the other hand, we have more H plus of hemoglobin attached to oxygen, other subunits will attach to oxygen more effectively and with higher affinity. It is cooperative effect. Guys, do you get the point about myoglobin and hemoglobin? Yes? Okay. Guys, if there was any ambiguity, you can see my email address in the first slide, and this is my cell phone number. You can be in touch through WhatsApp and text me if you have any question about these sessions that I told you, not about my colleague's session. Okay? Have my number, and if you have any question regarding these sessions, you can ask me. I will answer you at the first time. And no worry, study these again, and if you have any question, we can communicate with each other. Guys, when I'm telling about proteins with different subunits, we have something that increase the effect of that protein and something that decrease the effect of that protein. Or maybe decrease and increase the affinity. I want to tell you something under the name allosteric effectors. This is related to enzyme, but here I can explain you that tomorrow be more comfortable. Guys, do you know what does allosteric mean?
The attachment of the product of the protein to the same substrate increases the production of more of the product.
It is somehow correct. Guys, allo means other. Other part of the protein. For example, here is the place of attaching oxygen for hemoglobin. The allosteric effector attach another part and have effect on the function of these proteins. Okay? Other part and attach to other part and have effect on that protein function. Now I want to tell you about allosteric effectors. Here is a normal oxygen dissociation here. Guys, something decrease the affinity of hemoglobin to oxygen. These items shift our curve to the right. What does it mean? Could you tell me about the P50, about the affinity of protein or hemoglobin to oxygen? Decrease the affinity. And what is happening for P50? P50 was here. The place that your hemoglobin was saturated by oxygen by 50% of the ability. After decreasing affinity of hemoglobin to oxygen, what does the changes of P50 decrease or increase? Increase. You can see here, if I bound this part here, this is near 26. But if I continue it, it is near 30. You can and 3 BPG decrease the affinity of hemoglobin to oxygen. And near the tissues, hemoglobin can release oxygen more effective than in the lung. Okay, and in situation that H plus or pH increase and H plus decrease, PCO2 is low and 2 and 3 BPG is low, the R4 is stabilized and the affinity of hemoglobin to oxygen increase, and we have shifting of curve to the left. Okay? Is it okay? Guys, you can.
What causes the, I mean, like, what's the effect of allosteric effectors? Do they cause the right shift or left shift or both?
It depends. If your effectors like H plus increase, PCO2 increase, 2 and 3 BPG increase, they shift it to the right. But if they decrease, they shift it to the left. They are something that affect your protein function by increasing and decreasing. Guys, have you ever heard about going to altitudes? For example, climbing to mountain.
Yes.
In those situations, increasing of 2 and 3 BPG made to have lowering affinity of hemoglobin to oxygen, and on those decreased pressure above the mountain, our lung can work effectively. Okay? Did you get the point?
Yes.
In this another situation that just I want to tell you that on the liver that the pressure of oxygen is high, we have oxyhemoglobin. And oxyhemoglobin in R form come to the tissues and release the oxygen to the tissues, and after that get CO2 and go to the lung for exhaling that. Guys, I don't know that I should tell you about Bohr effect or not. Do you want or not?
Yes,
It-
that's important.
Example?
Here they
No, but I wanna
Then
know
why?
about.
No, very
we
good.
know it already.
Hmm?
We know it already.
You know Bohr effect? From where?
From school.
All of you know.
Yes.
Where were you school?
Congo.
Where?
That's good.
It's
It's
a
convenient.
UK-based board.
UK.
Okay, I'm not from UK, but we study from UK. Like
Cambridge
books
School.
and yeah.
Obviously, no. Okay. But I think all of you don't know all of you. Yeah. Guys, Bohr effect is a phenomenon that is based on the thing that happen between your tissues and your lungs. Okay? What is happening? Here, for example, in tissue, you had high metabolism, you had high glycolysis, you had high fatty acid, you need energy. All of the metabolism are, guys, if you are tired, you can stand up, have a rotation in the class, after that sit down. No worry about that, okay? Guys, here we have CO2 production in our tissues because of high metabolism. On the other hand, we have again increasing H plus and decreasing pH. Am I right? Because of high metabolism. Here we have an enzyme under the name carbonic anhydrase. Carbonic anhydrase that converts CO2 plus H2O to H2CO3. Carbonic acid. Okay. After that, these carbonic acids can convert to bicarbonate and H plus. What is happening now? Hemoglobin is working as a buffer. Am I right? Like protein has buffer. It releases the oxygen that bring from our liver, from our lung. Release the oxygen and instead of that, absorb the protons. On the other hand, by protons attached to each other, produce carbonic acid, and after that, both carbonic anhydrase again convert to CO2 and H2O, and we can exhale CO2. And now, here, the oxygen comes from our lung to our body and have exchanging of oxygen and H plus on the tissue and transfer these things between our tissues and our lungs. This is Bohr effect. Did you get the point? No worry about exam, no, I won't ask you. Maybe about carbonic anhydrase, just maybe. But I don't think so about it because I have just 6 questions from these 2 sessions. There are lots of things to ask. First, about disorders, we have protein energy malnutrition that can be marasmus or kwashiorkor. Do you know about the differences of these two?
In Kwashiorkor, your legs become bent like this.
Liver,
No, legs become bent like this.
why?
Okay, that I don't know, but your legs become like this.
Okay, just like
Maybe.
It has something to do with protein. In one case, the protein storage increases.
Yeah.
In another, it decreases.
All the body become fragile, but mostly the legs shorten.
okay. Guys, about marasmus and kwashiorkor, I will tell you. Marasmus is a situation that we have calorie restriction and protein restriction. Both of them. Okay, this is marasmus. And in kwashiorkor, just we have protein restriction, protein malnutrition. We have enough calorie. They have enlarged abdomen because of hepatomegaly, because of splenomegaly. Okay, it was about marasmus and kwashiorkor that is related to protein energy malnutrition. We have another situation that the folding of protein is destroyed, like Alzheimer's disease, like Parkinson's disease, like prion disease. I don't want you know about the details, but know that the Alzheimer is situation that the folding of protein is disrupted, and misfolding occurs. Another thing is about hemoglobinopathies, sickle cell disease, and thalassemia. What do you know? This is like about marasmus and kwashiorkor. I want to tell me about sickle cell disease and thalassemia. What do you know about them or about hemoglobinopathies in general?
Sickle cell anemia,
Uh-huh.
there's a big substitution.
Mm-hmm.
So
There is a
gluten
new type, new shape for it.
causes the shape of the RBC to become sickle cell,
Yeah.
and then that causes blood cells to clump,
Tiny shape.
and it can cause problems in the heart and other parts.
It's not a big substitution. It is just a little substitution.
One base.
It's one base. What happened? Deep effect. Guys, in hemoglobinopathies, I want to tell you. At first, they are genetic disorders, and in some situation, the structure is abnormal. We call it is qualitative. The quality of hemoglobin is changed. But in some situation, the hemoglobin structure is normal, but the quantity of that decrease. For example, in we have glutamate in 6 amino acids from N-terminal. Okay? This single glutamate substitute with valine. You can see here the codon. This is vowel base. Okay? The codon can be G, A, G. When the substitution occurs, the GAG constitute with GTG. Instead of A, we have T. And instead of glutamate, we have valine. A, basic glutamate, an acidic amino acid with negative charge substitute with a non-polar big amino acid. What is occur? The thing that occur, the RBC structure start to be abnormal and start to polymerize with each other, especially in the low oxygen pressure. You can see single nucleotide substitution that glutamate, instead of glutamate, we have valine, autosomal recessive disorder. The life expectancy decreased. It's not important to memorize just about glutamate and valine. You should know. Guys, you can see here that the beta chains attach to each other. And when we have problem in some situation that our blood has some, for example, branches, or maybe in small capillaries, they polymerize and plug and obstruct the blood circulation. And what does it occur? The big pain in different area of the body, the headache, the anemia, and something like this. Guys, in laboratory, we had a technique that put a drop of blood in slides and put another lamel on that, and we seal around the lamel. Okay? After that, because of decreasing the oxygen pressure, the patient that was sickle cell, the RBCs gradually convert to sickle cells. And we can see by microscope. It was great. It was one technique for detecting these patients. And about thalassemia, guys, I've explained to you that can be beta thalassemia and alpha thalassemia, that the globin chain have some mutation. Guys, I prefer stop teaching here because you are tired and my mouth is somehow facing some problem, and you are studying here, but indirectly.